Hi, I'm Trina Beckstead for Online Wellness Community. We're here at the 18th Annual A4M Conference. That's the American Academy of Anti-Aging Medicine. People with cutting-edge research, and one of them here is Dr. Stan Burzynski. Welcome.
Thank you, it's my pleasure.
Dr. Burzynski has an MD and a PhD and a lot of focus on cancer treatment. So let's find out what is the latest and greatest cutting-edge treatment.
Well, cancer is a genetic disease. Every cancer is caused by a combination of abnormal genes. It's a network, evil network of about 3,000 genes which are causing cancer. And to complicate the things, this network is different for every single patient. There are not two cancers alike. Everybody is somewhat different, like we are different genetically. In order to treat cancer in the proper way, you have to identify what is genetic makeup for individual patients, rather than looking simply under microscope and using the same procedure which are known for the last 150 years. We need to do what is called molecular profiling or genomic analysis. And that's what we do when we see the patient first time. We identify the genes which are causing cancer in the individual patient, and then we tailor the treatment plan for individual patient. It's like a boutique treatment. It's not like a Kmart treatment, it's like boutique treatment. We would like to prepare individual treatment plan. It takes some efforts, but if you do it right, the results could be astonishing.
What are some of the treatments that you propose then?
Well, we are using cutting-edge treatments. We introduce some treatments through our research, and we are best known for the treatments which are called anti-neoplastons.
Has some of the breakthrough in the genome, some of it is theoretical, but some of it is more science or is science? I mean, has that been helpful to you in cultivating these treatments?
No doubt about it. Well, every cancer has its own genome. There's a separate genome for breast cancer, lung cancer, whatever. But then for every patient, there's individual cancer genome, which is different for the genome of this individual. Well, perhaps in another 5 years or 10 years, when the patient comes to an oncologist, he will first study the genome of this particular patient, cancer genome of this particular patient, but it's still not possible. It will be too complicated, as we simply don't have medications which work on so many genes, like 3,000 genes, and we don't need it. At this time, it is sufficient to run, for instance, a battery of, say, 25 different tests, which will identify the most important genes involved in this particular individual cancer, and then select medications which would kill malignant cells which have abnormal genes. When we run such tests, then the test will give the answer and will tell us, well, this is the group of medication which is the best choice for this patient, and this is the group of medication which is not going to do anything, just give bad adverse reactions. So we select the right medications, we start the treatment, and in about 2 months we evaluate what are the results. In about 80% of patients who come to us, and they have very advanced cancer, we see substantial shrinkage of the tumor. And when we continue further, the tumor will go away. We have a substantial clinic. We are employing 22 medical doctors. We are located in Houston. And also we have a research institute, which is located in Houston suburb, which is Stafford, Texas, where we are employing also numerous doctors. We also have what is called pharmaceutical treatment, the pharmaceutical processing plant, which is making medication according to FDA requirements. We are involved in numerous clinical trials supervised by the FDA. Currently, we are involved in phase 3 clinical trials, which is the last step when you are trying to have marketing approval. I came up with the discovery of the medications which can kill malignant cells and which don't kill normal cells, which is something unusual. And chemically, these were peptides. I selected some of them, named them anti-neoplastons, and then proceeded through FDA approval process.
Thank you, Dr.
Okay.
Stan.
Okay, thanks a lot. Okay,
It
it's
was
my pleasure.
wonderful learning
Thank you very
about
much.
your techniques.
Thank you very much. Okay.